So earlier on when I was talking about debunking evolution with science, we can go through, I'm going to give you the short version and I'm going to put on this video. Let me start with this. Darwin's theory of evolution basically went into the DNA and the theory of mutation and speciation when today our technology goes through the DNA into cellular development, which shows things like machines such as this on the screen here, going up and down, pasting information on cytoplasm. It's a combinatorial problem now, and it's not an evolution thing, and I'm going to get into this a little bit here. This is called the C-ring F1 ATP synthase. And we're going to take a look inside of this thing. This is a machine inside cellular development. So I'm going to say that to start. There's machines. So I'm going to try to be real short here about it. So, and basically, we see all these highly productive processes. You see how this thing is being built right here, the DNA spine, and the proteins are being added in sequence specificity and are not random and not mutating or speciating, which that's another story. I'm trying to give you the short version. So let's move on to some of the hard meat of. And I just want to show. Here's a bacterial flagellar motor. When you see bacteria within cells moving back and forth, this thing actually has a motor. I'll say it again. There's a propeller motor on the back of these little bacteria strands, and inside the motor, you have about 14 parts. We're going to take a look inside this thing. This is called the bacterial flagellar motor. Here it is in a pictographical form. It's got about 14 different moving parts: tip, filament, junction, hook. It's got rings, rods, P-rings, piston rings, cell walls, stators. This is a machine. So when we're talking about evolution and mutation and speciation, we find the things that are building the cells. These things are little machines. And in science, the only thing that we could observe, which would be scientific, that makes machines is an intelligent mind. So let's take that point, and I'm just going to skip over all this amino acids and sequencing, because basically mutation and speciation theory is now being debunked in that we see here that the glycine, valine, cysteine, and histidine are all sequence specified, put together, versus chaos in the cell, where Darwin's theory of evolution stops, as in to build the proteins out of amino acids. What he's saying is, if you could look on the screen here, is this is what's going on inside the DNA, inside the genes inside the DNA, which is causing mutation and speciation. How you see how this alphabet board, everything is scattered, and it's not making any sense, all these letters. But what we're seeing now, what builds proteins and nucleotide bases, is sequence specificity of these letters to actually form information structure, such as sentences, but in cellular form, which is made up of hold on, I'm in my notebook here, so don't lose train of thought. These make chemical bonds, sugars, phosphates, amino acids, nucleotide bases. So let's just move it on here to the final pictures that I just want to end this. And I told you I'm giving you the real short version, and that is that basically, check this out. This here is a DNA strand. Inside of the DNA, and this is where Darwin's evolution theory stops, is he knows we have DNA, but he didn't get to see what's going on inside, what these little lines are. These are called genes. Okay, inside of these multiplicity of genes is the genome, which is a series of them. So right here to the right, we're dissecting the genome even closer. And what we're looking at here is these nitrogen bases right here are made up of adenine, thymine, cytosine, and guanine. Okay, but the problem is, and you see my little graph here, sugars are the blocks, the pentagrams, I guess they are the phosphates, and the nucleotide bases are the circles. Now, what's going on here is inside the genome, we see that in the middle, there's no... Oh, my phone's ringing. That's you calling. Okay, listen. Between the sugars and phosphates, you see these? At the nucleotide bases, there is no chemical bond here. You understand me? There is no chemical bond between the individual nucleotide bases. It's a separation. Okay? If there's no chemical interaction here, you cannot invoke chemistry in the sequencing, which undermines all evolutionary theory of speciation and mutation. This is the scientific insight that disproves the philosophical underpinnings of mutation and speciation of evolution using real microscopes and computers to look inside the genome, that there's no mutation here. It doesn't exist. I just killed evolution.